Okay, well, welcome back. It's the end of the semester. In principle, we have next week to do revised project presentations, but we're here. Lecture 15. The semester seems to have gone by pretty fast for me, maybe not so fast for you. I hope everybody is glad to be near the end of the semester, if not at it. And I hope people have interesting things planned for the summer. And I appreciate your patience in sticking out this class this semester. So again, the main thing we have today is people's presentations. I did send out a little worksheet. I would ask each of you to keep notes, just short notes on each of your colleagues' presentations, and type those up and send them to me, and I will anonymize them and send them on, so that everyone gets some basic feedback. And then again, what I've asked people to do is meet one group with one group. We actually have two groups and two individuals, and do a more in-depth sort of a 1 hour review and critique of your whoever you've been assigned to, and then write that up, send it to me, send it to the person you're working with. And the goal is, I realize we're on a short timeline, that everybody has done this by Friday so that over the weekend people can clean up their presentations and do a finalized version of the presentation next week. I'd hoped we'd do this a week earlier so we had a little more time to do it, but the goal is to be able to have at least one round of critique and review. I know that's not so common in classes in our department, but it's studied. If you're an architecture student, you do it. If you're an art student, you do it. If you're a media student, you do it. And I find it's helpful. I certainly do practice presentations and I get critiques and have do revisions. So I think especially for something like this, it's pretty challenging and complicated. It can be helpful. On the short timeline, maybe not as much as it could be, but something. So the video here is the results from that homework problem, just to show that it can be done. And we'll come back to that after the presentation. So the main thing today is project presentations, and then I'm hoping we can, if we have a little time, go over that delta notch example that I'd given as homework. And then if we have a little bit of extra time, I'll show you how to download and run SPML models. We talked about that very, very briefly at the end of last class, but we skipped over the slides. I showed the slides, but we didn't do it. And I think it is worth doing it together. So with that, we're at the student project presentations. We have 4 people to present. Who would like to go first? I didn't do a sign up this year. With 4 people, it seemed a little bit excessive to do a sign up. Any volunteer to be the lead on this? Or I'll just pick somebody. Since everyone, if you're not completely ready, you have next week, but everybody must present today. There's no outs on that. If it's not fully done, it's not fully done. What you have, you present. Alex, you're there. Let's start with you. Okay.
All right, I'm up. Okay. No, it's not going to work. Okay, can you all see slides? All right, so let's talk about not CompuCell, but kind of CompuCell. Meta presentation, let's go. So I've said this before, but so I was spending this semester working on optimizing the field extraction class, specifically working on parallelization of the various functions inside of it, rather than just like strictly trying to optimize existing serial code. which is a little different. So what is field extraction? So CompuCell has, I mean, it's running these simulations, so it has all this data in there that it's doing. So each time step, it's updating these various fields and data structures. But you can't really you can't visualize that yourself. Like looking at we could look at a giant array of information. That's really not helpful. So this is what turns the backend data into these pretty pictures that everyone sees. We turn into movies and we just render when we're just doing our testing and whatnot. So we need to pull that data out. But when you think about it, there's actually a lot of different fields that happen. So like in here, we have all the different cell fields, all the different cells, excuse me, and the borders. We could have just like a molecule field just spreading through that. So this is the same thing, same simulation, but different views. So we have this attractant spreading around, or individual cells can have specific fields specific to them. Like we can track attributes or other things. So these are all rendered in different ways, or they are stored in different places, and then we have to render them all together. Because like even all three of these require the cell borders to be drawn and that kind of thing. But it's important that this is fast and efficient, because the time we're spending doing this drawing means we're not running the simulation. But if we spend too long doing the drawing or the simulation, the UI feels sluggish, so people don't want to use it, which is not what's nobody wants. Then where does this slowness possibly come from? Because it seems like a really easy thing to do. But when you realize that these simulations can get pretty big. So like 250 by 250 voxels, even just that square area is 62,000 points that we have to look at. And then you have to think, we have to do that for just the borders, then the cell types, then any like fields in there, any additional things. So we're doing this 3 or 4 times at least. And then more if we have multiple graphs we're looking at the same time, possibly. So we have to pay that price. And then, so looking at the code, this is the original code. We just have this nested for loop. It looks at every single point in the map. If it's a cell, then it adds to this these data arrays, possibly doing memory allocations as well. So there's it's a lot happening. But we still have to do it no matter which way we do it. So then we look at parallelization. So because this is a single process architecture, it's not distributed in any way. The common thing is to use OpenMP, which is already inside of CompuCell and other areas of the code base. But so to go through everything of what is OpenMP, for those who don't know it, feel free to ask questions if I don't explain something well enough, because OpenMP is just great to know in general. So it is an API that enables multi-threaded programming inside of a variety of applications, but commonly people use it in C, C++, or Fortran. So it is important to know that these are thread-based. This is a thread-based thing. So you're not using processes. So that's good in some ways and bad in others, but the good things we care about is that we can still get that parallel execution across multiple cores, and then we share a memory space. So there's no passing of data around. We can all look at the same data structures and objects because they're all in the same memory area. But then we can still have private local variables for our personal state, for like whatever individual thing we're doing at the moment. So the way it works is you have your single thread running. I don't know if I can get a pointer, maybe not. So you have your single thread running, and then when you come to an OpenMP area where you want to do parallel processing, it creates a bunch of threads for you. So you don't have to call like fork a lot. Oh, there's a typo there. It's fork join. So this is fork join model. So you have this branching area, then you have your parallel code segment, and then once your parallel code segment is done, it removes all of those threads for you. So then you go back to single execution mode. So the way this looks inside of code, so you have a pragma statement, an OpenMP parallel. So as soon as you hit that point, you have multiple threads happening. So each thread will create all of these objects and allocate on its personal stack. And then this like shared thing here is saying that these variables are global, so everyone can see the same objects. And then inside of this parallel code, we can do lots of cool things. So two quick ones here. Obviously, if you're writing parallel code, you probably want them to work together in some way that isn't just doing their own things. So we have synchronization. So we can have like critical sections that guarantee that only one thing is in there at once. Or we can have a barrier. So it says everything has to stop at this point before we continue executing. And this is a hot topic, apparently. I don't really have opinions on. So the way OpenMP is designed is that you put these pragmas, mostly just these pragmas, inside of your code. But that pound pragma means it's a compile time directive. So the compiler could ignore it. So if you don't like link against OpenMP, your code will still run exactly the same, just serially. So it's neat that way, but also it means it's not as expressive as it could be. But we're not here to argue about this. So it makes the code look a little weird because you have these opening blocks rather than like function calls. But then, so we actually want to do coordinated computation. So we have the same for loop from before, but now we can just put this OpenMP4 on top of it. So now this whole for loop is now parallelized. So each thread is not executing the whole loop from 0 to this dimension variable. They each get a slice of that iteration that they work on. So then you can use this intelligently to say, once you store that output and then like some variable based on that, in this instance, we have j, but we have to store it as a local vector, which I'll explain all later. So then we choose not to like parallelize these inner functions because OpenMP will already be using all the threads we have allocated for it to do that outer function. So we usually don't gain anything from trying to like do nested parallelism. And then, so lastly, some other neat things you can do with OpenMP is if you want the code to do, you want your threads to do multiple things at the same time, but they're all different. So they give you these sections. So I use this repeatedly to, a lot of these functions have various data structures they need to store data in, because this was for edges, so edge points, but like, so yeah, this is the plotting the cell types. So you need to store all the types, where all the types are as Cartesian point fields. And then you need to store that in a data structure describing what these look like. So that's how VTK works, but lets you do a lot of this. Rather than doing 3 allocations serially, you can do them separately. So it's kind of handy. Now let's look at how this all got implemented. So let me switch to code now. So this first function is for doing, we never looked at any 3D stuff in our simulations. Did we? Well, there are a few in the demo samples, but so this was like the biggest offender on time killing because you don't have 3 dimensions to loop over. So this one was an interesting function. So like, so this, it tries to optimize. So it will iterate over all of the cells, but this was not parallelizable for technical reasons I'll get to later on. So, so the left is the new version, the right is the old version. So because we know ahead of time how big this field is going to be, we can do all this allocation at once. The same thing happens before. But then we just simply have to do our for loop over the outermost loop. And then all of this happens across like 8 or 10 threads. That's it. It's really easy to convert. So there's a little more. So because now we're allocating this ahead of time, we can just set the specific point, which is what this was doing already, but this has an implicit, maybe we could do allocation. So we want to avoid that, but it still works. So the other, get to the other function quick. So this one that we did look at a lot more is plotting the 2D cell types in a vector. So this is interesting because when you think of those plots, we had a lot of non-cell space, a lot of medium. And I mean, black is black, you could render it, but you could also just not. So because of that, we don't know how much we actually need to render, which makes it kind of complicated. So we can't say, let's just render the whole thing. That actually is too slow, surprisingly. Slower than doing it serially in a lot of cases, but that's on later. So here we have, each thread will then store the points it decides to plot internally. So the original one just skipped those because it was doing this allocation each time. Maybe VTK does things under the hood, but it didn't. It had this implicit knowing how much was there. So we can then use this essentially the same code, and then store all of the data points for each voxel along with their type. But because each thread has this local store, we have to combine them together in the critical area. So we don't avoid, I mean, you're going to get pointer crashes and whatnot if you try and do it without this, because you can't have the kind of shared access to data structures. And then we can collect everything now that we've gotten all of our points, so we know how much we need to render. And then we can use that to allocate all of our memory inside of VTK. And then do another for loop, because now we know how big this array is and where it's all going. We can simply do that in essentially pre-computed all those positions and do another parallel for loop. So that's how pretty much all of these functions broke down. Some of them got a little more complicated in how much they wanted to render, but they all essentially look like those two versions. So that made it surprisingly easy from when I first started looking at the code to change to rewrite everything, because a lot of it looks the same. So then how well did this work? So some of these functions turned out really well, especially the 3D ones. So when I did this testing, I was running on 10 threads. So those really big 3D rendering fields saw like 7 and 9x speed up, which is almost the perfect scaling we want. But some of them didn't do as well. It's like, so these one, most people use the 2D rendering, but so the cell field, we didn't use the hex version, but the Cartesian is only a 2x. Filling the borders, it's just a little more than 3. So when you're running 10 threads, you want close to 10x. So why did this happen? And then most of the reason is this contention thing. So we need these critical sections because we don't know how much we want to render ahead of time. And then I did testing, and then rendering everything in these fields was actually slower than the serial version. So we couldn't do that. So the threads are stuck trying to merge all their data together, which is, we can't do much about that unless we can somehow, unless somebody's really good at this stuff, maybe there's a way I couldn't think of to combine it all. And then secondly, depending on which computation exactly we're looking at, we have this memory bound thing. So really what we're doing is just a giant mem copy when you think about it. We have the data structure in CompuCell, and we have to convert it to the VTK version and store it in a different memory location. But so this was the just the coordinate or the field, the cell type fields. So that's all it's doing, is that looking at the data type, storing it in VTK. But if for the border one, which actually got a slightly better performance, it is doing a lot more work per data point, because it has to look at all of the neighbors and see, are they the same type as me? So it's doing more work per point. So it's more compute bound, just a little bit. So we see some interesting stuff there. And then the other big trade-off is how much do we want to, what do we want to optimize for? Do we want this case where most of the cell field is black, so it's not filled with cells, or this one where it's mostly filled with cells? So this one this is the bottom one is where we would use that coordination, but also critical areas, because we aren't plotting the whole field. That's what I went with, because at least most of the simulations in class didn't fill up the whole area with cells. So I ran into one issue I couldn't solve. So there are a few instances where the thing we're looping over is not an integer, like the for loop for I in range, whatever, the classic thing. CompuCell uses a few iterators, which is normal C++ thing, but the supported version of OpenMP in the Windows Visual Studio C++ compiler is on like version like 2.1, 2.2, which only does those integer loops. The latest version of 3.0 does support complex data types, but is not implemented in Visual C++. So just have to wait for that support to happen eventually. But then the next steps, which I didn't get to this semester, is the possibility of using GPU acceleration. So I did a lot of digging into VTK, and it is smart enough to use a graphics card for the rendering. It does that for free, actually. It has a complex class structure, and it knows how to put, it looks at the setup and it knows what to use, which is nice. But but you can't give it data on the GPU already, as far as I can tell. But there's definitely the opportunity to do this kind of rendering or do this kind of field extraction on an accelerator. And at that point, it would probably, I'm sure it would be faster to render the whole field and skip any kind of coordination, because the GPUs just have so many more threads available. So that's everything I got.
Questions? How many people have used OpenMP? Anybody? And anybody used MPI? TJ. Well, I know TJ has. I mean,
So
no.
I guess that's a good question, because I just don't know about everyone. Are they mostly biology people? Or I know Joel said he did a lot of coding, but
I actually have a question. OpenMP then will do all the managing of starvation, concurrency, everything automatic for you?
Sort of. So concurrency, yes. It creates the threads and assigns the work for you. Starvation can be, so there's you can do, this is the only example I have. So when you're doing this, like a for loop, you can tell it how to schedule that information. So like you can tell it, there's a dynamic version where it just says if a thread needs work, then it gives it more. And it dynamically assigns which like J to give, or you could have it statically where each is given like a fixed chunk ahead of time.
That's a great tool. I didn't even thought that would exist. I was like thinking with myself, it must be a pain to just think about everything and hard code everything. It would take.
Yeah, because this has been around probably 30 years or more. Same with MPI, but they're really great tools.
So the load balancing is partly automatic then?
Yes.
You said some of it, but you ran into places where where you had problems with that, right? Where the synchronization was chewing up your speed up.
Yeah, that's the functions that needed synchronization saw the worst speed up, yes.
I mean, this was the problem in building the core parallel POTS algorithm, because it's stochastic. And so you need to make sure that each thread is operating in a region of space that can't affect any of the other threads, because you're actually doing writes. You're not just doing reads, you're doing writes. And so you have to be very careful not to have collisions in your writes.
Yeah, the POTS code is interesting because it gives everyone like a subsquare of the field to work on each worker thread.
And so it depends on the fact that no object, one of the reasons medium is a null none type is because if any object is big enough to cross those squares, then the parallelization fails, because you can have two threads writing into the same object. And so you have to be guaranteed that no object, there's a buffer. You have your threads working on this square. You create a barrier of 8 squares around it that nobody works in. And then supposedly the squares outside of that are safe. But if you have a really big object, it could run over that barrier, that safety margin. And when it's interesting. So very early in this class, or actually I think we dumped it from this year. In the past, we've done the very old and simple soap bubble growth simulation, where you run the simulation with no volume constraints. So some cells disappear and some grow. So at the end, you have one huge cell. So that breaks any parallel algorithm, because as the cell gets bigger, no matter how many, you could use fewer and fewer squares at the same time. And CompuCell crashes. It doesn't actually crash. It just does very, very strange things because you're doing this conflict. So I think this is fascinating to see this. I really appreciate it.
Yeah, I've had a lot of fun with crashes because a lot of this was just writing C arrays, and VTK isn't like, the documentation is there, but it's very verbose. So it didn't really, like, understanding the data layout took a while, because if you don't have the data layout right, when it goes to render, it would crash a bunch. Those were fun to figure out.
Now, when you said that the VTK has GPU support, is that there but turned off in CompuCell, or what are the issues that I know you already did a lot for a semester, but if you were going to continue to GPU support for rendering, what would have to be done?
So when it has VTK has GPU support internally, so you give it the data, and then it I only saw, my understanding of this was from like a random blog post, I think, not like documentation. But VTK looks at the local setup either at compile time or runtime, I'm not sure. And if like OpenCL is available, it will use that. So OpenCL is this open source implementation of like a graphics driver, like functionality. It will use that to do the rendering pipeline. It has special like implementations of all the rendering classes for that. So it will choose those rather than the CPU version.
So that depends then on TJ. What would have to happen? I don't see CompuCell chewing up my GPU on my laptop when I run it.
So when I can confirm this as best I could at the time by like, I would see CompuCell using like 5 or 6% of my GPU.
Okay.
The local version I had built, so I knew it didn't have like, it wasn't using the GPU version, the GPU code inside of CompuCell. So as far as I can tell, it was VTK doing it.
Fascinating.
That's right. It's just that I think typically the visualization that we do just isn't a very big load for, you know, any GPU that's been made in the last 5 years.
Mhm.
Yeah, I mean, doing, you know, car arrays is just not a heavy burden for a GPU.
Well, certainly the CPU side of it is expensive. You do 3D rendering in CompuCell. It takes a long time.
Not for long.
Well, that's
Yeah, I don't remember the number, but like I mean, it's like the most modern GPUs, you have several thousand CUDA cores like on NVIDIA ones. So even if you have a rather large 3D image, it just chews right through it. It doesn't really care.
well, if we could get that working, that would be great. TJ is buying us an A100, so we'll see if that
Okay.
helps us out.
Nice.
Of course, that doesn't help. No, we don't know what hardware CompuCell will be running on. People might be running on a 10-year-old PC too.
Hopefully they'll have upgraded since then a little bit, but who knows?
But that's great. Other questions? This is very different from what we've talked about in class, but very important.
I don't have a question, but general comment as someone who's not a computer science individual by training. I thought that you presented a lot of the high level concepts in a very agreeable way where I could follow what you were doing and why you were doing them. Definitely couldn't have done it myself, but I just wanted to say that because I thought that was well presented.
All right, thanks.
I agree. I think given that we hadn't talked about any of these issues, I really appreciated the time you took to explain some of the basics. And the issues are tricky. As you know, in the end, the code looks simple, but I know getting it to work is not at all simple. These are challenging problems.
Yeah, it is, at least for the rendering stuff, it is rather simple as in like algorithmically, there's just a lot of it. You have to understand all these, all these moving parts you have to get right. That's what complicates it.
I mean, do you run into, I mean, I know you had to use some criticals and other locking functions. I mean, did you have problems where you were having collisions between threads, which you had to address, or was the code structured already in a way that basically that was where collisions were less of a problem?
No, it was, there were a few. Find one quick. So yeah. So you should be able to see Visual Studio Code. So this was the, we didn't really use these in class, and I don't know how often they're used, but like each, like voxel and then each cell can have like a vector assigned to it. This was the cell version. It's like just like a XYZ pair, not a location, but just like a pointer kind of thing. So it needed to ensure that each cell was only used once, only like was being rendered once. So that required a lot more. This was the one that performed the worst, like with the speed up, because it is like each thread is tracking which cells it's seen, but then to combine everything, it needs to compare it against what everyone else has done. So that just takes a long time, and you have a lot of waiting.
So when we do things like isosurfaces, is that are those all pre-existing VTK calls, or do we actually do that computation? I know when we calculate the surfaces in 3D, that's time consuming, but I don't know if that's done with VTK or
what is, I don't know what an isosurface is.
I don't
So-
think Alex didn't go here in our code
No.
base, so I'll answer that one. Yeah, so that one is generated by VTK. And that would be one that where you would feasibly see your GPU sweat a little bit, because VTK uses either a 2 or 3 dimensional float array, and will calculate isosurfaces based on the data and then whatever, you know, optional specifications you provided.
So just for some background there, I mean, we've done it, as you pointed out, Alex, we didn't do that much in class using 3D arrays. And mainly that's because they take longer to run. And so you can get the idea usually in 2D. And if we're doing in-class exercises, we don't want to spend 10, 20 minutes waiting for the simulation to run. Now, when you're looking at chemical fields, in 3D, you have a problem because how do you see the field? It's a color density. So what do you render? And so there's an option in 2D also that draws isolines. It'll draw a contour where the field is 10, a contour line where the field is 5, a contour line where the field is 7, and so on. And that we've used once or twice when we're doing chemical gradients. In 3D, that's really the only way to effectively represent concentration fields. And so what you'll get are things like tubes, where instead of being a line, it'll be a surface.
I did see those because the code for like pulling out that data is in this file, and I worked on it.
Yeah.
So like to test it, I created some simulations. So and you have these weird, like, you just get these like wavy lines and stuff that just go across the whole field.
And that generating those in 2D is not so bad, but in 3D, you then have to have a meshed surface that represents those. And that's a fairly expensive calculation to do. The other one that is something is, again, in 2D, cell boundaries we usually draw. In 3D, when you actually show the boundaries as well as the volumes, that rendering can be relatively expensive. Again, I actually don't know how CompuCell does it. I do know that part of the reason that the precursor to CompuCell was written is that the original code I wrote 30 years ago, I did all those calculations by hand. And the graphics library that I used was discontinued. Sun had a graphics library they used. And when we were faced with rewriting the entire rendering package with the new library, we decided to throw the whole code base out and start over and do something more modern, rather than to hand calculate all of the boundaries and surfaces in 3D. So that's part of what happened, actually, was that the library disappeared. So when you mentioned that the Visual Studio doesn't support the latest version of OpenMP, I thought, uh-oh, that's a...
I couldn't find much info on it, but they are like working on it. They're not refusing to do it. Just Microsoft takes time on some things.
I think that's pretty fundamental. A lot of people depend on OpenMP, so it's and it's an interesting question if people work with MPI versus OpenMP. Having shared memory makes things a lot easier.
But then it's a trade-off. It limits your scaling. You can only go to one machine.
Sure.
So like some, depending, I mean, if you go in really big scales, like some will use both.
I mean, that algorithm for the pots, that checkerboard algorithm for the pots works in MPI, And in fact, we had an MPI version. We had an MPI version of the precursor to CompuCell. 20 some years ago, 25 years ago, full MPI version. But it comes back to something that you mentioned that I remember vividly learning from Jeffrey Fox, which was that parallelization of this kind works best when you have a lot of computations per message pass or per synchronization.
Yeah.
And one of the problems with that core POTS algorithm was that you didn't have that many computations per message pass. And in the old days, when you had a multi-box architecture, the latency for doing the message passes was pretty high. And so you spent all your time sitting around waiting for your other box to send you your updates.
You would have to have like just a massive field to make it worth it.
These days with faster interconnects, if the cluster is designed well, then those latencies aren't so bad. But actually the scaling was pretty good. It was just slow. I mean, the linearity and number of boxes was great. Number of processors was very good, but the speed up per processor wasn't so great because you spent all your time waiting around to synchronize.
Yeah.
So seeing this done is really exciting. It sounds wrong to say, well, I've been waiting 25 years to see this kind of thing, but to see it done again, it's really nice. I appreciate it. So is this likely to find its way into a release, TJ?
Oh yeah, this this will get integrated with some other work for release, most likely in our next release.
Okay. Other questions, comments? I mean, in a sense, this is, in one respect, this is easier than some of the other things not to do, but because the documentation, you have to document it for developers, but the user should never see it. From a user's point of view, this should be a speed up, but it should be completely transparent.
Yep.
But if you could help, can tell us a little bit about, explain to us what we learned and how we could do better. And if there are things like particular data structures that CompuCell uses that could be rethought to make parallelization easier.
Yeah.
That would be.
Yeah, I have a question to that effect. So typically, when doing this sort of acceleration, whether it's just with multi-threading or throwing in some GPU support, this is sort of like the point that you've taken this is sort of like the end of iteration number one, of many future iterations. And typically after doing this, the next sort of step is to then reevaluate the structure of the algorithms and reevaluate how the code itself could be restructured to better support exploiting parallelism, which is especially complicated when you aim to target various architectures, ideally mixed architectures. And so I was curious about, as you hand this off and, we go to integrate it and continue developing it, if you had any suggestions or recommendations about, the sort of next step of what could maybe be, what are the biggest weak points here, to maybe try to eliminate in future code development.
I hadn't spent too much I haven't really thought about that too much. And we can talk. We're going to talk on Friday.
Yeah.
Yeah, so I can come up with answers by then.
I don't think we weren't trying to put you on the spot on that.
Oh yeah. No, it's just, I thought of it when, when you were going through some of the types of visualization that are not as readily parallelizable. It was a lot to do with neighbor calculations. Not having all the information beforehand. And so I guess if anything, which points here are the weakest that maybe the code needs improved to support better parallelism.
Yeah.
Yeah. But yeah, we're going to talk on Friday, so we can dive into that.
And there was another point you made, which I also think is important. Maybe more for people who are going to go on and do code development. And that your comment about it works better when you have a lot of computations per message pass was well taken. The other one was the point where you showed the example where you had the cells in a clump in the middle with medium around them, and a simulation where the field was filled with cells. And you pointed out that the optimum parallelization might be different in those two cases.
Mm-hmm.
And so one problem that we face with CompuCell, which isn't faced with a hard-coded environment like PhysiCell or some of the others, Simune, is that we're trying to be a completely general purpose environment. And so people, in fact, may use it very differently. And so the load balance may be very different. One person might have a single cell surrounded by medium in a huge lattice. Another person might have the entire lattice filled with tiny little cells with a lot of surface on it. Somebody might be doing lots of chemical fields. Another person might not have any chemical fields. Or somebody might have a lot of subcellular models. Complicated. And so there are times when we've experienced this where we've tuned the simulation to run in a particular situation very well. And in other situations, that tuning actually makes the performance worse. And I was thinking, you had that example where you pre-sweep to do a calculation and then you parallelize. And I was wondering whether one could do something a little bit like that, where you look at the code, say, what fraction of the lattice is occupied by medium? And then make a decision about which algorithm you're going to use downstream for that. There is this function which we never turn on called box watcher, which more or less tries to do that in a very naive way. It says, is there in fact a sublattice which has cells in it where everything around it is medium? And then I draw a parallelepiped around that and ignore everything outside of it. If you have a single cell surrounded by medium, that increases your performance enormously. If the lattice is completely full, it doesn't do anything. And one could do a smarter algorithm, but I think that was an important question, which is, how do you optimize when you don't know the deployment?
But then, I mean, there's a trade-off with the code base wise, because now you've just doubled your code to support those, like in that instance, those two cases. So there's a trade-off in one direction, the other. Yeah.
Well, one place where the code becomes very complicated, it's the main reason I think why people don't do a lot of developer work in CompuCell compared to other environments that are somewhat similar, is the fact that we support all these different lattice types. And so having to be able to support 2D and 3D, square and hex, periodic and fixed boundary condition, arbitrary neighbor range, you wind up having to write methods for all of those different cases. And usually for your particular problem, you're going to use one lattice. And then writing a solver for that one lattice is pretty easy. And you don't feel like going back and having to write 12 versions of it. So that sometimes that flexibility certainly comes at a cost. You hope it's hidden to the user, but it makes it harder for the developer. So I think your point was well taken on that. Sorry, Joel, do you have another question?
Yeah, actually, I have a question about, from my understanding, the way CompuCell works is every step, there is a certain amount of possible changes that a cell can take, right? So
Right.
in that case, isn't it easier to assume which are the possible changes we can have and then write an algorithm that is a better account for just the change instead of the whole system?
You possibly. So like one way you could do it is to, I mean, you could track every change. But then if you want to do, you're doing that in parallel, so you still have the contention overhead of that like collective log of changes. So theoretically that might not be any better.
Right. I was thinking on avoiding those cases and just ignore the cases that don't change. That might be enough, almost like a YouTube algorithm for for scaling videos, which is taking account things that change and compare them to things that don't change. And the things that don't change, you just let it be. And
so, the issue is that you still have the data isn't stored in two different locations. Like the CompuCell has its data structures, and then VTK has its data structures.
oh, got it.
So you move
Okay.
it, you still have to move it from one to the other. Like I guess you could keep everything in the VTK format. I mean that's its own complication.
Yep, you're right. I didn't thought about that. It's two different types of data structure.
I mean, there was a GPU only version of CompuCell at one point, but that had its own issues. It's tricky. Again, that flexibility comes at a cost, and Connor has been experiencing that painfully. The early versions of these programs actually. The inner loops that I wrote, again for the solvers, not for the visualization, did a lot of netting out of things. So we tried to avoid computations as much as possible by sort of pre-computing what didn't change, exactly that kind of thing. And that worked really well, speeded things up a lot, but then you lose flexibility because there are situations in which it doesn't behave. And so thank you. That was great. Any other questions before we move on? Thank you so much. Please make your comments and send them to me and pass them on. It can be just good job too, but if you have suggestions, ways of improving things, that's great. Thank you so much, Alex, for being the lead on that. Maybe Connor, having talked about you, maybe you should go next.
Sure. Yeah. I think Andrew is going to share screen for us today. All right. Awesome. So we'll give him a second to get.
Yep. Can you see my screen? All right. Okay.
Okay. So as Professor Glazier alluded to, this is not quite the presentation that I thought we would be giving today, but it should be an interesting one. So we're going to be talking about our work trying to replicate the origin of multicellularity. Next slide, please. So just a quick overview as a whole. we talked about this a little bit earlier in the semester, but we're going to give some biological motivation for the simulation that we're working with, a summary of the paper, and then most of what we did in CompuCell is pretty similar to the paper, but we're going to dive into some of the details that turned out a little bit differently. And from there, we'll finish with next steps, extensions, and conclusions. And you should feel free to interrupt at any point. Yeah, you're good now. All right, so biological motivation. Next slide. So this is not a biologist, but this is a quote that I think of a lot when it comes to multicellularity, and you'll see why in a second. So this is Walt Whitman. He was a poet, and he has this poem called Song of Myself, where one of the sets of lines are, do I contradict myself? Very well, then I contradict myself. I am large, I contain multitudes. And that's pretty applicable to how people think about multicellularity. So if we go to the next slide, there is an apparent paradox when it comes to germline versus soma, right? So sure, we're reproducing, but when it comes to individual single cells, having this divide in cell differentiation means not all the cells get to reproduce. So there's kind of this question as to why a collection of cells would end up in this state. And here we have two examples. One is digastelium, the slime mold on the left. And when it's in pretty strenuous conditions, it will grow into the stalk, where the cells in the stalk will die, and it will release spores to reproduce from the top. And then on the right, we have Volvox, which is a type of green algae. And here we have its asexual reproduction cycle. And once again, you can see in the top left corner of the graph, after it reproduces, the somatic cell undergoes senescence and death. So there's this big question of how we're supposed to approach multicellularity. Next slide, please. And one possible thing to keep in mind is that, within the biosphere, there are both unicellular agents that have been very successful and also multicellular agents. So there should be cases where we're able to explain why we get the evolution of one and not the other. And typically, even though it's a pretty common characteristic of a lot of multicellular agents to see this germline versus soma divide, it's an evolutionarily derived state in a conglomerate of cells. So we need to go back even earlier to get at the story of multicellularity. And that kind of begins with when single cells start becoming integrated and having a shared fate, possibly even before they have that degree of differentiation. Which is interesting because a lot of models will kind of presuppose the advantages that we see with multicellularity, which means that we don't actually get to look at kind of the emergent strategies and capacities that we get when conglomerates of cells begin working together. And we also don't get to explore as much the types of ecological conditions that would select for that kind of behavior. Next slide, please. So this is the paper that you heard us talk about earlier in the semester, Evolution of Multicellularity by Collective Integration of Spatial Information. And before I pass things off to Andrew, next slide, please. I want to talk about a couple of remarkable features for this model. So something that Professor Glazier has mentioned multiple times is that when we're doing simulations, we don't get anything for free. We need to specify it and include it. But this paper walks the line a little bit because what it declares is kind of a space of possibilities where something like a multicellular conglomerate can emerge under the right conditions, but it isn't deterministically presupposed where regardless of the state of the simulation, it will always turn up. And in that sense, unicellular and multicellular success are possibilities. And based on the mutation of ligands and binding sites, which we'll be talking about a number of times during this presentation, there's the possibility of both multicellularity and also speciation within a single CompuCell cell type as they select to have different binding capacities through individual reproduction. So that will bring us to the dynamics of the environment in due time. But for now, I am going to pass things off to Andrew.
So our model parameters to go over, that we have our chemical gradient, how that was constructed, and the persistent migration, and ligand binding, and then finally, not shown here, but the evolutionary algorithm. So in constructing the chemical gradient, we have a 500 by 500 lattice where a focal point is determined randomly within this lattice, and the maximum distance from this focal point is going to be one of the corners. And so by choosing the corner with the largest distance, the rest of the gradient scales linearly from that distance all the way down to 1. And then to make the gradient noisy, we have a 10% chance for every pixel to be 0. And finally, this gradient changes locations in every season, and so every season another focal point is chosen, and then the gradient scales based off of that. And so in chemotaxis, within the paper, there is a vector where inside the cell, the chemical gradient has a center of mass, and this vector is constructed with the cell center of mass, and that is the direction of chemotaxis. Whereas in CompuCell, the direction of chemotaxis is constructed from this equation, where it's based off the specific concentration, and it's incorporated into the minimizing of energy algorithm. And then there is persistent migration, in which every tau p steps, a new location is recorded, and a vector is constructed from the last time it was recorded. So that way, wherever the cell is moving, there is going to be a bias in that direction from the previous vector that was constructed. In the ligand binding, there is a genome for which is dictated as a bit string, zeros and ones of 52 length, where the last 6 are of the ligands of the medium, and the rest are cell-to-cell binding parameters. And the strength of the binding is determined by the Hamming distance. And that Hamming distance goes into this distance function, which is then modified through the size of the whole vector, and a parameter to just increase the overall energy, starting from 4 and ends at 52. And if there is a medium to medium, cell to medium binding, then there is a piecewise function where the energy of that is determined by the specific index of the ligand to the cell's receptor. And so based on the index, there can be value of 1 to 4 and 0 otherwise. And so for evolution, after every season, every cell has the probability of reproduction that changes depending on how far away it is from the focal point of the gradient. And after reproduction, each ligand receptor has a 1% chance of mutating, which is just flipping the bit. And then the population, as it has already grown past the original starting population, it gets each of the cells are randomly culled back until the original population size has been obtained. And now I'm passing it on to Connor.
Yeah, so I'm gonna be describing some of our process to try to replicate this and things that we've had to get a little bit creative about. And so Andrew and I will bounce back a little bit, back and forth for this section. Next slide. So the first thing we had to implement, which CompuCell didn't have, is that there's no such thing as a patchy gradient where we set things up in CompuCell normally. So that was something that we had to initialize, and the way that we did it was that we built a simulation pretty similar to the one that we did in homework 7 with the swimming lanes, and we had a cell of changing volume chemotaxing up this patchy and noisy gradient. And that was our attempt at initially trying to get at some of the general findings of the paper, because a multicellular mass is going to chemotax in some way similar to a cell that has a larger mass. And making sure that we could replicate that initially. Next slide, please. Another thing that gave us a little bit of trouble was that in order to have those ligands and binding sites that Andrew was talking about, we had to pull in a new plugin called AdhesionFlex that allows us to declare those. Whereas normally when we would do mitosis, we would clone the attributes of the parent to the child cell. We aren't able to do that exactly when we have a bit string of ligands and receptors. What we have to do instead is that we need to directly call the parent adhesion vector from that AdhesionFlex plugin, randomly mutate it without changing the last 6, because those are the ones that correspond to the medium and are the same throughout the original paper. And then we need to assign that to the child cell before moving on from that point. So this does work. It's just a little bit more complicated than the way that we've been using mitosis otherwise in class. Next slide, please.
And so ideally, for the contact energies, we would like to implement these two formulas. The first part, which has the definition for J-celled cell, and the second condition is cell to medium. Implement them conditionally, but within the AdhesionFlex plugin, it has muparser, which is basically speeds up the formula that is used to bind. And so at the bottom, there's no ifs, ands, but or lesses about it, meaning within this specific the rules of the syntax in CompuCell's implementation of muparser, we weren't able to use like the C++ ifs, ands, or lesses. Fortunately, we were able to do not equals and greater than. And so what we ended up doing is multiplying the truth value of the specific molecule's density with the formula. And so if there are specific values that we've assigned within the XML file that allows this parser to identify or to return a value that evaluates to true in these conditions. So if it's if the molecule is a 1 or a 0, then the final condition is the final condition will be met. But if one of the molecules is of the medium, then that's what the first two statements are for. First one is for the first molecule, and then the second condition is for the second molecule. And what was implemented here is that in order to identify whether it's of the medium, these molecules were initialized to be in between 0 and 1. So if the molecule is in between 0 and 1 and the second molecule is not, then we can implement this formula where we have to multiply the value of molecule, let's see. It should be molecule. I guess we haven't multiplied it by 10, but we're supposed to multiply the value of the molecule by 10 so that way we can get the integer value. And then that will be sent as completely summed up into the as the total binding energy for each cell to cell or cell to medium. And now for Connor.
Right. So something that we've been alluding to has been that adhesion flex is not super friendly with these circumstances. And as you could see from the type of operator we were having to use to replicate their equations, it was more us kind of forcing adhesion flex to do something it really was not comfortable doing, to say the least. So we have a large number of molecular species per cell, specifically 52. And on a local version of CompuCell, it takes about 2 minutes to run 10 Monte Carlo steps. Now in the paper, every simulation in order to reach an evolutionary steady state requires 40 seasons, where the shortest season length you need to see multicellularity is 40 times 10 to the third, which gives us about 16 million Monte Carlo steps to even have a chance at seeing multicellularity. And as you can see, this translates eventually to about 6.2 years for us to actually get the simulation to where it would need to be. So this was not something, well, we can do the next slide first. So right now we're saving your life by not running this code, and that's why we haven't done a demonstration yet. Originally, Andrew and I had made a plan where we would be ahead of schedule for running the code, but more complications ended up popping up, and none of those, even if we had been ahead of schedule, we could not have been 6.2 years ahead of schedule. So we have some steps left that we need to do. We need to honestly probably set up a new plugin specifically for this kind of situation, which was not something that we had been thinking was necessary to begin with. We also might try to do a simpler version of this simulation that has fewer molecular species, because frankly, if it's going to take 6 years to run on my computer, NanoHub is never going to finish. All of this, though, was discovered last night. But so next, if you go to the next slide, you can see this is literally the 2 minutes into the simulation, just because I wanted you to be able to see how it was coming together. And if you'd like, we can walk through the code some once we're done walking through kind of like what we're planning on doing. But as you can see here, we did manage to replicate the chemical gradient the way they specified it. We do have the 200 cells initializing randomly throughout the field. They seem to have inheritance and mutation working properly. So really most of the stuff for this simulation is in place. It seems like most of the input to output mapping is correct. What is not correct is the actual computational process. And for that reason, we cannot promise you that this definitely does the exact same thing as their simulation. Yeah, next slide. So like I said, for trying to get the simulation shape for NanoHub, we really need to take the time to do an additional plugin, which is going to be a little bit of a time crunch, given where we're at. And we also are considering the simpler version of the model. That would also be a time crunch just because it would be more parameter balancing for a lot of complex variables. I was trying to work on part of one this morning and have not made a ton of progress yet. So those are first key steps. Besides that though, next slide, please. Some extensions that we had talked about with Roland, who was the PI for the paper, was that in the original simulation, they looked at the trajectories of single cells and cells in multicellular conglomerates through space. But what wasn't explored was the trajectories of cells relative to the clusters that they were in. And that seemed like something that could be done with the base simulation that had not yet been explored very thoroughly. Next slide. So something else we talked about was that in this simulation, cells don't consume the gradient. They just move along it. So we discussed the possibility of making it so the cells can consume the gradient and get full, and that how full they were would influence the strength of chemotaxis. And this, if we paired it with our mapping of cells relative to their positions within clusters, could be interesting, we thought. The final extension on the next slide that we were considering was that the same way that the possibility of multicellularity versus unicellularity was opened up as a possibility but not predetermined, there would likely be ways to extend this style of simulation to see if you could find ecological conditions that selected for germline soma division, which seems like it would be a productive extension. I think that would be the more complicated one out of the extensions that we considered, and probably the one that we have the least progress on outlining right now. But that brings us to our conclusions as far as the general talk, and then we can show you the code for anything you'd like to see. First is that this paper really does take a novel approach to multicellularity compared to a lot of the other work that we had seen. And we do think that we managed to replicate the key parts of the code, but the built-in plugins are not sufficient for how we need to implement it. So that's something extra that we're going to have to take on to see this through to its end. And yeah, our current code may take 6.2 years to get at multicellularity, but we still did it faster than Mother Nature. So we're going to count that as a win. Yeah. So I don't know if there's any general questions for the talk or comments or grievances people want to share, but then we can also pull up the code and go through that for whatever people want to see.
Well, I have a couple, but I'd like to see the code too.
Okay.
I mean, I wondered one thing was, did you write a simulation of that first one where you have a gradient with noise, and you do something where you plot the velocity of the cell as a function of the noise amplitude, or the size of the cell, or lambda chemo, or the gradient strength?
we had a
Because
simple.
that by itself would be a nice demo on NanoHub where the user can select the noise amplitude, the gradient, and see how fast the cell moves under those different conditions.
So we did work on part of those. What happened was that we ended up part of the chemotaxis that we were having difficulty with. So we switched gears until we had covered chemotaxis in class, and then implemented here. So we do have part of a simulation that does do exactly that, that it has a single cell chemotaxis and Apache gradient. And that would actually be really easy to brush up and finish in a way that would be good for NanoHub. It's not
because
Yeah.
you could do a parameter scan where you plot the velocity as the function of the parameter. And then the user can select, say, the noise amplitude or the size of the cell and see how those change the behavior. I think it could be quite valuable just for a single cell.
Yeah, we could very easily finish brushing up our rough draft code of the single cell and get it to do something like that. So that's an option for NanoHope, given that the current state of the simulation is looking a little bit incompatible. Does anyone else have questions on the presentation, or should we start going through the code?
So this is the adhesion flex plugin where we have 24 adhesion molecules, 24 receptor molecules, and 6 medium receptors, which these are actually not to be like interacted with. So those have a value of -3 compared to the other ones, which are 1 or 0. And basically they don't fit any of the conditions, so they'll just return a 0. So we have to initialize all the pairings. And then this is our formula. And then finally, in the adhesion matrix, we just multiply it all by 1 because the formula gives us the strength that is part of the paper.
Yeah, so when it comes down to it, it's not pretty to look at. And part of that is that it's not quite meant to be used in this kind of way. This is a creative use of adhesion flex that does not accurately represent how the documentation encourages it be used. Yeah, Joel.
Did he actually code everything on the paper as XML?
Did they
The
code
original
everything?
paper? Yeah, everything that you did, they did also in XML.
That's a good question. I haven't actually. They use. I think they did it in. Does the package that they use C? That's what I thought. Yeah. So they didn't do their simulation in CompuCell. That's part of the they used the cellular POTS model, but it wasn't in CompuCell, and that was part of the reason.
It was their own unique implementation, right?
Yeah.
Okay.
So I mean the adhesion flex setup, and just to give people like maybe some background for how it would work typically, Andrew, if you scroll up just a little bit. So here it's declaring the density for every molecule that we initialized for the medium and the cell types, which in our case is just the medium and cell 1. And then what would usually happen is that, and you have to declare some density for all of them at that point. And then if you go down to the binding formula, typically what would happen is that you would have something like molecule 1 plus molecule 2, or you take a min or a max given the relationship between the two of them. And we ended up turning into a logical operator to try to approach what they did in the paper.
The new parser has its own specific set of syntax that will work. Some some of it is particular to C++, but also some logical statements don't work as expected.
Yeah. So Professor Glazier, if you remember from the time you and I met like a week ago, how the AND operator crashed it, and then we went line by line and isolated that. The same thing happened with the less than sign. And that strikes me as very odd.
I wonder if it's not that the XML parser, because view parser doesn't expect it to be an XML.
Mhm.
If the XML parser isn't somehow looking at a less than sign as an opening tag,
Yeah.
and then crashing it. I wonder, I don't remember, does somebody who remembers XML, does ampersand have a special meaning in XML? So I'm wondering whether the problem is that somehow it's being parsed as the wrong language, essentially.
That would make sense.
Yeah.
Now I might have expected that greater than would have the same problem because it thinks it's a closed tag. But maybe the parser only looks for closed tag after an open tag, and so you're all right.
I truly do not know.
Yeah.
We figured out this through brute force and
Okay.
changing the rules one at a time until we found out which ones broke it and which ones didn't. And once we saw that less than broke but greater than didn't, we just translated the formula.
But my guess is that if less than is breaking it, it's because it's being parsed as a tag open. I don't know if you could put the whole thing in double quotation marks or something and tell it don't parse this, and I'm not sure that that's what's going on, but because clearly taking all your logicals and turning them into floating point multiplies is going to slow life down a lot. Although it still might be pretty slow, but that's interesting.
I actually would expect that to be the case where it's expecting an opening tag, because the error that is sent doesn't seem to be an error with new parser. When you send in syntax that doesn't belong to mu parser, it would say that it is from mu parser and that there's like a recursion error.
Well, the miracle of Google. Ampersand, double ampersand is a reserved character in XML.
Well, that's one mystery down.
Yeah.
Stack Overflow says use ampersand AMP, semicolon ampersand AMP instead of double ampersand. Now, of course, mu parser may grok on that.
Right.
Somebody else had this particular problem.
That's fascinating.
Yeah.
So
But-
it is. So my guess is all three of them somehow are XML parsing errors.
Yeah,
Oh, well.
that makes sense. Well, so this formula does technically seem to do what it's supposed to, as much as we can tell given the speed that it moves at. If you go down to the adhesion matrix, just to finish explaining how it works for people who haven't seen it yet, this creates a symmetric matrix, right? So if you specify molecule 1 to molecule 2 being like adhesion 1 and to receptor 1, then receptor 1 to adhesion 1 in the reverse direction will also be specified. And the binding parameter is a multiplier for whatever you get from the binding formula. And that's how adhesion flex is meant to work. But in our case, it's all ones, and then it becomes a logical operator. And then the rest of the interesting stuff is in the steppables file.
Yeah. I guess I can talk about the initializing seasons function.
Sure.
Oh, sorry, just before you do that, it looks like you have to look up how it works. It's not a triple ampersand quotation mark. So there's a command, an XML command called cdata, which tells it not to parse what's inside of the argument.
Mhm.
So you'll have to look it up, but maybe you could get it to work.
Okay.
Again, it may break through parser, but it does seem that this is a known problem.
Mhm. So in our initializing season function, because it's a linear gradient, there's just some parameters at the beginning for slope, y-intercept, and then how big this the gradient is depending on the size of the lattice, and then also the probability of the having holes in this gradient. So first, there is just a little section to identify, or if the starting location hasn't been identified, which is on the first iteration, it will just randomly initialize it. Otherwise, no, yeah. So the reason is here it's just it's just initializing it to zeros, because if it hasn't been initialized, it's just none. And so we just identify the 4 different corners and find the corner with the largest distance, and then that becomes the maximum distance. And then for every pixel in the cell field, we compare the x and y coordinate of the pixel to the starting location, and we scale that value according to the distance, and then just through a binomial with a given probability. This is a 10% or 90% probability that it will be the chemical field, and then 10% for there not to be a chemical field, just be 0. And then in the mitosis steppable, we are calculating before the season changes, we are identifying how far away we're identifying how far away cells are away from the focal point, and we're comparing that with maximum distance. And so we basically use the distance from each cell to that focal point, and give it a probability of giving it a probability from 0 to 1, depending on that distance, the normalized distance. And if it returns true, then that cell gets to divide. Otherwise, it doesn't. And then we are updating the attributes, where each child is receiving it receives the adhesion vector, and for every element in that array, there is a 1% chance of flipping. And yeah, that would be, so that's the initializing seasons, and then the mitosis steppable. And Connor? No, Joel?
I actually have a question. It might be because biology is not my forte, but So the way you establish the gradient is the noise is the lack of gradient in a specific point.
Yes.
Does that translate to the biology of, do you see that in the same sense of in nature and not like a mix of the gradient?
So biologically, I can imagine like if we had some sort of a pile of chemical or food for cells, they will be there. There is a large concentration in the middle, and then that concentration decreases the further away from that focal point. And actually, in the beginning, that intuition is what we used to implement on the gradient field. But when we kind of went back into the paper and we looked at the details, it really is about just the 10% of there not being a chemical. And the whole point is because having those holes in the lattice actually makes it more difficult for a single cell to move up, as opposed to comparing it to starting all the way at the end, where it actually is a lot more difficult, both in terms of intensity in the gradient and higher chance of there not being the chemical. And with the multicellularity, when you have higher surface area, it's more likely to actually overcome those holes. And then that's what the paper is testing.
So Andrew's totally right that like this is how it's implemented in the paper, and that's their reasoning for doing it. This is an idealized field in a lot of ways. For starters, like if you have, and so it's a static field throughout the lifetime of a cell, right? That lasts an arbitrarily long period and then suddenly vanishes, reappears somewhere else, establishes a new gradient across the field. And I think that the type of noise you would expect to see would actually really depend on a lot of biorealistic details that we don't see in the simulation. For example, is the chemical being poured in slowly? Like, why is it that the cells just encounter this discrete transition? You would also, like the way Andrew and I were thinking about doing originally was that you have more holes the further you are away. Because you're not close to the source of the gradient. So then if something messes with the field, you're likely to see more issues with it. You could also make it more realistic by saying, instead of a discrete, at this random point, there's no chemical, you say, hey, there's a chance that the slope changes at this random point, that whereas there was this much chemical here before, now there's 1/3 the amount or something like that. So all of those are things that could be done to make it more biologically realistic, but in the sense that the goal of this was to initially replicate, that's not what they did.
Yeah, I was just trying to understand, nothing like to criticize the work of you guys. It's just more like for me to understand what is the idea behind the noise, and have you tested to see if the cell get like lost track of the chemotaxis after a specific hole in the, you notice like if it's connected to the center of mass of the cell, or it doesn't have any relationship?
So we haven't tracked specifically in terms of like where in the cell, but because of the size of the cell, it's as a size of 5, like, and that's a large portion of a single hole is a large portion of this whole the cell's surface area. So it actually does make it difficult for the cell to move up the gradient, and oftentimes there will be moments, or there will be instances where individual cells don't actually chemotax up the gradient, because in any given instance, there might be a chance where if it stochastically moves in a direction up the gradient from a global standpoint, but in the local perspective, it actually is decreasing in how much of the gradient is actually underneath the cell. So it actually wouldn't do that, and it would get stuck.
Well, maybe that's another thing. If you're going to do your single cell example, changing the persistence time of the motion also
Mm.
Because the longer the persistence time of the cell's movement, the more likely it is to jump over a hole.
Yeah, that's true.
And so that may be important. So if you could adjust the sort of the amplitude of the noise, the amplitude of the gradient, the size of the cell, the persistence time, then you should be able to see how often does the cell get stuck, how far does it get per unit time, these kinds of questions. So I don't want to rush too much, but we will want to get through everybody today. Does it run? Even if it takes 2 minutes to run 10 monochrome steps, does it run?
It
runs.
does run.
Can you show it run?
Yes, we
Sure.
can.
Sure.
I mean, it would be nice to see the evolutionary algorithm too, but I think maybe save that for next Tuesday, because I want to make sure everybody gets to do at least one.
Okay. Change that. That isn't.
You're gonna have to. Are you running the right simulation?
Don't think so.
Okay.
I'm going to pass this off to you. Yes.
Okay. Yeah, that sounds good. Okay. Can everyone see my screen player? Okay. So it'll take a moment to initialize, but right now it's establishing the randomly located 200 cells and the gradient. And I can hit play, and in momentarily you'll see it go from start to 0, and in exactly 2 minutes you will see it go from 0 to 10. But no crashing. And then if you'd like, next week what we can do is that we can remove some of the features of the simulation just so you can see the seasons changing. Actually, that's something that we can show you if we transition to trying to make our NanoHub application the single cell case. You'll actually be able to see the seasons changing in that simulation. But so also something that, well, the rest of you wouldn't notice this, but Professor Glazier, you'll notice, which is that when the 10 steps come around, these aren't going to explode or turn into weird stripes. So the even though adhesion flux is slow, it does seem to be producing an acceptable contact energy.
Well, I did discover something working with Josh, which is another adhesion flex annoyance, which is if you change the parameter values of the adhesion flux in the start function, they're reset to the XML values when the simulation goes into the step function.
Okay.
If you put the initialization, in the step function, you have if MCS equals 1, then overwrite, then you're fine.
Yeah, that is what, so what we have is, well, I'm not going to pull away right as we're about to get to this exciting 10th step, but we initialize it within outside of the start function and in the adhesion flex steppable. So I feel like that should probably avoid that issue.
Yeah. But for some reason, the values of if you create the cells in the start function, and then you write, give them, this is the amount of chemical 1, this amount of chemical 2, and so on. But it then gets the step function, it rereads the values from the XML and puts them back where it was. Which is the XML is supposed to be executed before the start function. But for some reason, the XML initialization for this particular plugin gets called after the start function. And I'll tell you that was another. I've used the adhesion flex for simple things, but these were both things that. That was actually Josh was the one who figured that one out. I did not. We knew there was a bug, but boy, it was not easy to track that one down.
Yeah, I mean, on the upside here, so
Yeah, there it goes.
It's at 10, and something you can see is that the cells look a little bit bigger now. That's because when I initialize them to prevent overlap, I initialize them at a slightly smaller size. So this is them reaching the target volume for the simulation. And if we stay here for another 2 minutes, you would see it get to 20. But I am willing to bet that no one wants to stay here long enough to see them chemotax.
I'm afraid that would be slow with this,
I think so.
but I keep thinking there, as you say, I mean, there ought to be a way of getting something similar to work without it being quite so elaborate. But and actually implementing the algorithm that they used would be easy. You know, you could, if you opened up the plugin that does adhesion flex, there's a place where you put the evaluation, and if you knew what it was going to be instead of having to run the mu parser, my guess is you could actually do it pretty simply and pretty fast, but you would have to write a custom plugin.
So in that case, should our plan be to try to get a single cell version working for NanoHub? And then once that's done, we can always come back to this and try to get it in better working order?
Yeah, I think so, because the problem with putting in a C++ plugin to NanoHub, well, actually, it's not, as far as NanoHub is concerned, every app is the same. But you would have to CompuCell, which is I mean, not horrible. I mean, poor Alex has been doing it a lot, but it's not the most enjoyable exercise in the world. I mean, the whole point of CompuCell was to not have to do compilations, and so adding that plugin it's not terrible. Giuliano would know how to do it, for example. TJ certainly knows how to do it. You get Maciek's help if there are issues. But again, if you look at what's happening is that if you look at the loop structure, and this is the innermost loop in all of CompuCell, which is evaluating those energies, which is, if you're 1,000 by 1,000 by 1,000 lattice, that routine is called 10 to the 9th times per Monte Carlo step. So it needs to be fast. And what it does is it iterates over every neighbor twice, because it doesn't net out non-changing things. And then within that, it iterates over every pair of possible species without checking whether the species are present at all or whether they're the same. And so you're doing essentially the maximum possible number of iterations that you could possibly do. There's no intelligence in the plugin that says, I don't need to evaluate this branch because nothing is going to change. And again, if you want to write a completely flexible plugin, you could fundamentally, almost any optimization you do that way will break under some condition. But in the case that you have, where you have pairwise interactions, you should only be iterating over the pairs. You shouldn't be iterating over the non-existent off-diagonal terms in that matrix. And with 24, how many did you have? 30, how many species?
We had 52.
52. So instead of doing 26 evaluations, you're doing 52 squared evaluations. And putting that in your innermost loop. Plus, you're doing floating point multiplications instead of logicals. Although I think that C data will get you your logicals back. So that might be worth a shot to see if it helps. But I think fundamentally the problem is you have these loops that are spending most of their time doing nothing.
That makes sense.
But that's great. Okay, well, thank you. That was great. Any other comments or questions I told people that you didn't have to worry about being too short today. The trick will be getting through everybody. Who wants to go next? Josh or Joel and Jax?
I guess I could go.
Okay, go ahead.
So mine's not quite finished just yet, so hopefully it'll go much quicker. But my project was on emergent networks, particularly a slime mold simulation. It was based on a 2011 paper by a undergraduate named Nicholas Simler. So a real quick biological context. So the main stage that we're focusing on is the plasmodium stage of the slime mold. During this stage, it's considered an amoeba-like organism with broad tubular networks. The slime mold will create networks to communicate between different food sources. It's a lot like how ants, when they find a food source, they'll kind of leave this scent trail to communicate to the other ants to, follow the path of least distance. And so kind of branching off that, the network is constrained by the environment. So the number of food sources, if there's any obstacles such as physical barriers or toxins or anything. And so the main kind of mechanism that the slime mold uses to create these networks is it creates these actin-myosin fibers, and then it uses this passive transport system to transport the chemicals that it uses to create the fibers, as well as the food that it actually collects. And this flow of plasmodium steers the fibers that are being created. And so again, the main kind of computation that the mold does is creating these tube networks. So it's very important that the tube development has some rules. There's three main ones. The long, thin, and open tubes are generally eliminated. Tube size has an upper bound, and the more plasmodium that flows throughout the tube increases the size of the tube to that upper bound. And so as far as the framework, he originally used the cellular POTS model, CompuCell 3D, but it was the older version. So that was one of my main tasks, was porting it over to the 3.4.x. And so the main kind of CompuCell 3D plugins that he used was secretion, diffusion, and decay, as well as chemotaxis. And so again, this is kind of the model in terms of creating these actin-myosin loops to create the tubular network. So the kind of flow of data is that we load in kind of the simulation volume in the form of a PIF file, and we essentially just set the general structure of the environment, because that's the main constraining factor. So in this simulation, we essentially only have food sources and obstacles. And so the food sources secrete an attractor, and the obstacles secrete kind of repulsive things. And so that's kind of the main framework for how the direction kind of goes. As far as movement, again, it uses this autocatalytic loop, and it essentially just transforms it from a blob structure to more of a net. And when we run the simulation with the steering panel, you can actually change a certain parameter that is very prevalent to this phenomenon. And then another kind of thing that is relevant to movement is that low cohesion and secretion of attractants together restrict the volume and connection. So it's a connected network, but it can, throughout its optimization process, it constantly is pruning these connections. And so it uses volume constraints as well as connection constraints to do that. And going into that, the fitness, it is expected that the slime mold achieves optimum structure by oscillation and growth through the autocatalytic loop. And so the main thing is that growth and oscillation have been modeled to increase and decrease the volume. And this is essentially all the same things except the minor changes. We had the chemicals saturated at a specific level to mirror finite molecular size, and food sources had to increase the amount of attractors that they secreted during fixed intervals to appear less homogeneous. Oh, sorry. So as far as the results, like I said, I have gotten the simulation running. I just haven't, I wanted to do a couple more things like getting the steering panels and everything fixed, but there's three main kind of layouts, the PIF files. So there's this 4 food sources. This is the paper result on all these slides. He has 3, 1 with the slime mold starting in the middle and 1 starting at one of the food sources. I actually haven't been able to find a difference between NL and MST. They're both kind of this structure, but I'll hopefully figure that out by next week. And so that's kind of the end of the talk. So I'll go to my code now. Sorry, it's doing the thing again where it won't just let me share my desktop. Hold on. Oh, I think this might. Okay. So this, I believe, is the 4 food sources with barriers, which I think I forgot to include. But it's essentially a little bit bigger. So it's essentially the red is the food sources, and blue is the barrier, or I called it wall, and the green is the slime mold. And so when we run this, we'll just kind of randomly walk out. But again, there's this attractant field, so you'll see that all the food sources are secreting this attractant. And then this lambda value, if we change it to 0, you'll see it creates that blob-like structure. So this is a pretty important parameter, so can mess with that. It was around 300. So this is for the repulsion, can change that. It just now it's being attracted to the wall because the lambda is much higher. So we generally don't want that. So it's usually a negative value. I haven't been able to, the connectivity is a parameter that determines how because if without this connectivity being so high, it can kind of disperse and go into all these little blobs. But I haven't figured out a way to get that connected with the steering panel yet, so this does nothing at the moment. But in theory, once I get it working, if this was a very small value, then it'd be really not tubular and broken up and not one thing. And so as far as to-dos, I guess I just need to update the presentation, and with all of the, I just need to run all of the maps is what the author originally called these different PIF files. And then kind of, he did some graph analysis with GIMP. That's all he said. I don't know what he did. So I think I might skip over that. But that's all I have for now. I'll hopefully have more next week.
Well, that's a lot. I mean, it might be nice if the user could sort of say, throw down 5 sources or X sources and X obstacles. And so instead of reading it from a file, the user could generate some quasi-random initial conditions.
Yeah, I was looking into that. I messed around with just like, originally I wanted to like integrate a GUI where you could just like drag and drop things, but I started looking at the source code and like how I would, either make a new, like embed a new like graphics window or anything like that. And I've never used VTK or anything. And so I was honestly thinking about making a tool just like something completely separate, just like a simple Python GUI with like tkinter or something, or PyQt5, I think is what you use. And it'd be fairly simple to just make a GUI where you can drag and drop it and then click a button and it'll automatically generate a PIF file. So I briefly thought about doing that, but I wasn't sure about how I'd integrate that in NanoHub. But I could try if that's something you think would be very helpful or useful.
Well, I certainly wouldn't try to create a new GUI interface. That's way, I mean, you'd be most welcome to do it, but that's way beyond the requirements of this class.
Mm-hmm.
Generating a freestanding app, we've done that, and we actually, CompuCell used to be released with something called Cell Draw that would let you do that.
Mm-hmm.
People didn't use it much because to get a drawing package to be good, you have to do a lot of work, because the commercial drawing packages are very good.
Mm-hmm.
And so you wind up having to reinvent a lot. I think for this purpose, if you could throw down N sources and N sinks, and
Like in
maybe,
the steering panel.
yeah. So what I suggested what I said at the beginning was where you just have a feudal loop that watches for one of the steering panel items to change.
Mm-hmm.
So you say, okay, you have a pull down, which is or a slider, which is how many sources, how many sinks, maybe whether they're square or elongated.
Mm-hmm.
And then you have something that's a button that says start simulation. So you select those two, and then you hit start simulation, then it draws it, and then it reads the other things and continues from there.
Mm-hmm.
It's not the most elegant thing in the world, but it works within the constraints of the steering panel as it's implemented.
Okay.
Which is why I put that little example in last week's lecture. If there any confusion about it, I'm happy to show you how to do it. But that winds up being pretty easy, and it does give you the ability to then create an initial condition, because normally the problem is that there's no the system doesn't wait for you to create an initial condition, and CompuCell doesn't give you the option of sort of type your value here, hit return.
Yeah.
And so you have to force, if you want CompuCell to wait for user input, you have to force it to wait.
Mhm.
And so that, but I think that's pretty nice. And it looks, I mean, it looks like it works really well. I mean, you could imagine doing a little bit more, as you say, more analysis of the connected graph and this kind of thing. It may be on the connectivity parameter. It should be, if you go into the, I have a question, if you go into the XML, if you run this now, run it now, while it's running, if you go into the model editor and you change the value in the connectivity plugin there, does it do anything? If you change that to 5 to, you know, can you, does it let you change 5,000? Make it 0.
Yeah, it looks like it. It's breaking up a little bit.
Okay, so it is steerable. The issue is, and I'd have to look it up, some of those XML parameters have to be set and get in rather complicated ways. There's somewhere where there are Python methods that are built in that make it easy. Sometimes they're a bit hard to, and I don't know how to rewrite that. The fact that you can steer it from that panel means that it's possible somehow, it should be possible to overwrite it from Python.
I wasn't, because I do the attractor and the repulsive lambda. I use the get XML element in Python, but I couldn't. I tried that with the connectivity, but it needs like some like the lambda, the attractor lambda is like the lambda parameter in the, what is it? Yeah, in the chemotaxis plugin, the, like there's a lambda parameter, and then I set it, you know, get attribute and then the attribute dot lambda.
Right.
But I wasn't able, there's no, for the connectivity, it just says type slime mold and then penalty 50,000. So, well, I guess now that I'm looking at it, maybe there's a dot penalty attribute, but
no, it won't be a dot penalty attribute. You'll have to do get XML element. You'll have to do the get XML element thing.
yeah, well, that's what I meant. Like get the XML element and then access the penalty from, yeah, because I wasn't sure how to access the penalty from, or can you see the code right now, or is it still just my, okay. Yeah, so this was the code that I had, and I kept getting this weird, I'll see if it'll do it again.
Ironically, for the chemotaxis ones, you don't have to do that because the chemotaxis has got methods written for it explicitly. So you don't have the old syntax. We had to use that get XML element all the time, which is a huge pain. And for the ones that people change a lot, we put methods in so you didn't have to do that. Non-type set connectivity strength.
Yeah, so I think it's this this connectivity local flex plugin, which I didn't see. All I saw was a connectivity. I didn't see a connectivity local flex, so I just assumed that that was maybe the same thing.
What does it do if you put that in? I don't know. Well, that's in the XML though.
Yeah, well, it's already there. So if I
Right.
if I try and do it again, I just get an error saying that it's
already. Yeah, well, it shouldn't be. It's not a local flex because it's a global parameter. So but in the Python, I don't know. It may not be. It may not be swigged. So let's see in your let's see your Python reference to that just for a second. I don't want to take too much time because we want to make sure Joel but let's take a quick look. And so where are you trying to get? So if cell
So yeah, originally I thought it was maybe the cell was none type, but then I realized that that wasn't.
No, it would be a get, it would be that get XML element.
Mhm.
And so you'd have to do, could see if you could get XML element for the argument. Just get XML element on that. And why don't you do a dir on it? See if that works. So let's see where.
Oh, I think this is it. Oh wow, there's a lot.
Well, because I think it's doing the dir on get XML element, so it's returning every possible XML element call. It should see if the connectivity has a method associated with it.
Like,
So
what if we just
I mean, this is a place where looking at the C++ probably would be easier because we should have we find out what the interface is for it. But yeah, I don't know. I don't think that'll exist, but you can try.
yeah.
Yeah, so it isn't. There may be, typically what you have to do is you actually have to walk that get XML element, you actually have to walk the parse tree. So it'll be get XML element, there'll be lots of square brackets and commas with nothing after them and empty quotes.
Okay.
But I haven't ever tried that one. That's one if you email Maciek, he can give you the code for that in 5 minutes.
Okay.
Or he
Yeah,
basically
I'll figure it out.
you can't change it. But there is I would go to Maciek directly. He's the only person I know who for whom the parse tree syntax is natural.
Okay. All right. That sounds good.
But it's unfortunately it doesn't have it's not going to be available as an attribute like that.
Mhm.
Because for that to exist, they would have had to implement it specifically.
Mhm.
Joel, did you have an idea about this?
No, unfortunately not. No.
If you look at changing the say lambda chemo old style, you'll see what the syntax for those parse tree operators looks like. But I would definitely ask Maciek directly. I'd send him the code. You have his email address, right?
I don't think so.
Let me. I think it's also supposed to be possible, and this is not something I've done much, is I think you should be able to, in the XML, name the argument. And then that name should be available inside of the Python.
Just give it an ID.
Yeah,
Oh yeah, that's what I did too with the other ones. I did it as well for
and
the
that doesn't work.
No, that's how I
Okay.
use the get XML element. To get it, you have to have assign it an ID so it knows
Okay, fine.
where to grab it.
Okay. So I sent you and Maciek an email to introduce you.
Okay, yeah, I got it.
Okay, great. And so
Yeah, thank
he's
you.
he's usually super responsive.
Hmm.
So I would send him your code, explain what you want to do, and he should. The answer may be you can't, but if you can, he'll send back the code that you need.
Okay. All right. Thank you.
Okay, sure. Okay, great. Okay. Other comments for Josh? This is a great demo. I'm looking forward to having that up on Nano. Joel, I guess you're on. You and Jackson are on.
All right. Jackson, do you want to start showing that slides?
Yeah, do you mean yeah, I can pull the presentation up.
Cool.
Can everyone see that?
Yeah, we can see.
I just click slideshow, right? I don't use Google Slides though. There we go. All right, so our goal was to create a model that could model all the different types of cell death. So there are multiple different types of cell death. There's non-programmed, which is just the necrosis, which
Oh, there you go. Never mind.
all right, there we go. Sorry. So there's programmed and non-programmed cell death. Non-programmed is necrosis, which generally occurs when some form of like trauma or like a toxin is introduced into the cell. Programmed cell death, they're split between apoptotic and non-apoptotic. And so initially our goal was to get a majority of the cell death types in, but we'll talk about the complications later on. We wanted to focus on pyroptosis and necroptosis originally. Pyroptosis is an inflammatory type of cell death that is involved in a lot of bacterial infections. The paper that we used to try to find our model was a paper on pyroptosis and its connections to the COVID virus from the pandemic. One of the issues with pyroptosis is that it has an inflammatory response. So once it undergoes the response, it releases particles that actually increase the immune response and actually make the infection seem much worse for the host body. Necroptosis looks extremely similar to necrosis and actually was not fully recognized as a type of cell death until the mid 2010s. It's initiated like all other programmed cell deaths, which is generally through some molecule binding to a death receptor on the cell and then initiating the mechanisms. One of the issues we initially had was trying to find literature on models of different cell death and trying to find the rates. And our goal is to create a unified model of all the different cell death types because we wanted to see how the different mechanisms and the different pathways for cell death could interact with each other. We also wanted to see if there was a certain point when we would if we were able to run the simulation that one type of cell death would overtake the other type of cell death and like what different factors cause that. And the main goal was that we wanted to have a better understanding of cell death signaling and its function in the immune system. So we chose to use this single mathematical model of COVID induced pyroptosis, and also an incoherent feed forward loop for necroptosis. And then
Yeah, I can take over here because the other part is in a different thing. So if I can share my screen, just a second. Yeah, I wrote most of it in a different tool. Now I cannot find it. Just give me a second. It's not showing on my screen here. Huh. Just give me a second. There we go. So the replication of the pyroptosis model was a success. Everything is working according to what they report. After a couple of iterations, I found the problems that I was facing, that I was reporting earlier in our classes. But basically, it's still following the same concept that we saw on the first week, the workflow for developing models. We start analyzing the representation, the biological representation of this model. And after that, we go for the mathematical representation of the same model. And here we start understanding a little bit of the rate in which things are created and generated inside the cells, right? So we technically saw this a couple of weeks ago, but the difference between describing as an ODE and actually describing as a arrow with the rate of change and what that implies. So here I try to do the thing that we were seeing in class of like representing a whole arrow with this specific equation for it, and not in the ODE modules, so we can trace back and forth what is happening inside that molecule chain. Hold on a second, it didn't load. Same thing here for the other molecules. So I did this. This will be available for everyone that is interested in C and want to read a little bit more about it. So I did all these representations here showing what each of these ODEs were representing on the first image we saw at the beginning. And here's the fun part. This is the antimony code showing the same equations that we saw at the beginning, the rates of decay, the rates of which things change from one state to another. And here is what we can see. This is, oh, I don't know why that did that. Hold on a second, let me just restart here and clear everything. Okay. Yeah, here is the concatenation of all the things present in this model at once. So it's a little bit messy and it's hard to compare with what they have on their paper. But we have this option of simulating each thing, each protein at a time. And here we can see that happening. And for example, the NF-kappa B, we see that is the same rate. The only difference here is that I think they're using, these were basically divided in 8. So a 2x4 image. So the tendency for matplot is to squeeze a little bit of the plot. And that's why it kind of looks a little bit different. But if we look up the numbers, it's exactly the same model. It's doing exactly what they're describing here. Same for the production of NLRP3. We can see that they have the same rate. And again, because of that squishing of the plots, we see a little bit more here than here, but we can actually get every single number if I do a for loop and get every number at every point in time here. We can see that it's exactly the same numbers that they are showing there. Same for everything. So this is a way for us to confirm that the model that they wrote in their paper is actually the same model that we are using here, is comparing which what they have as a response to their model, if we got the exactly same representation here. And I am glad that finally I got this week and everything seems to work fine, which goes to the other part of the project, which is implementing everything that we saw there into the CC3D model that we learned this week how to do it, right? After you have a antimony model working, so the rates are all in place, we can run. The sad news is that I didn't have the time to go over everything because it's a lot of details that you have to keep in mind when you're doing this. There is a lot of molecules and each one is responsible for a thing in this model. For example, we're going to see this is basically our first implementation of the CC3D, and all the values here are all arbitrary. But now the goal is to put everything that I got right from the paper here on this model. And here we can see what is happening. There will be a video in the end too, right, Jackson? But since we have a live model, I just think I could play. The thing that we're trying to show here is the difference between cells that are actually infected by virus load and initiated by pyroptosis. This model is just considering pyroptosis. We know that cells die different ways when they have a viral load. Most commonly, they go through a apoptotic type of death, depending on the virus. If the virus shut off, for example, this defense mechanism of the cell. And there are other more complex things involved, like interference, that also react to a load of virus. But this is a simple model, just taking in consideration the paper and extrapolating a little bit on this part of the model, which is how neighbor cells will react to this information that we have about pyroptosis. And here we can see the first, it's named DAMP because that's how they used in the paper. But this actually is, we are trying to represent here a viral load and how it spreads. It might be spreading too fast. We should probably check literature to see how much it spread on a mean, on a medium. And here we can see just the, as we saw in the model, each cells, when they go on the pyroptosis, they make little holes on their membrane to liberate this molecule, IL-1 beta, which tell other cells in the neighborhood that something is wrong with them. They're infected and they should be prepared in some way or another, and also signal to the immune system that something is wrong here. And we can see as the simulation goes that a lot of the cells actually die by the pyroptosis signaling and not so much by the viral load. The green ones will be cells that are actually starting pyroptosis by the virus load. So this is true representation if we look at what happens with people that get COVID, for example, where the actual inflammation and the cytokine storms is what kills, because the immune system sometimes reacts too strongly to this signal. And we can see that if the signal is strong enough, it will overtake the viral load. So it's a very intelligent, on quotes, intelligent way of preventing the virus to spread to different cells, but it comes with a cost of a bunch of cells dying because of the signal that they're getting. So yeah, I can get back to you now, Jackson, if you wanna. Let me just stop the share. I don't know how. Sorry.
It's just the video left. I mean, if the simulation was shown, do we need to show the video?
No, it's okay. We don't have to.
Because yeah, the only thing left is the video and then questions. And I mean, the video is just exactly what we just saw.
So what happened? You weren't able to get Hoffman's thing to replicate, right?
He never responded to my email asking for code.
Yeah.
So-
Oh,
And I haven't had time enough to explore all the possibilities for changing the distribution to be exactly the same as the one they had. And
find it yet.
yeah,
I've just been putting in numbers. So
I wouldn't worry too much. You did a lot. Getting this to work already is interesting. I think you're right that the diffusion lengths of those two molecular species are important within that simulation. But the fact that you can do you validated it and you can get it running inside of CompuCell is already a lot. And
right.
this issue of what's called bystander effect, which is that when cells die, their neighbors die, is very important. Chemotherapies don't work very well, but if there weren't bystander effect, they wouldn't work at all. Because cytotoxic chemotherapies kill some fraction of cancer cells, and you depend on the fact that your immune system and this kind of triggering leads to cells next to cells that are dying to die. Now, of course, if it runs away too much, then you have one cell in your body die and every other cell dies because of the signaling. So you don't want that to be too much of an amplification. But that kind of amplification is essential for preventing viral spread, bacterial spread, and cancer. And so what turns it off again is something that has to be thought about. Why don't you get this runaway cell death always? There has to be some anti-inflammatory signal that comes in and turns it off, which isn't included in the base model that you have. It's correctly not included because it's not in the source. But I think, I mean, I don't know, Jackson, what your plans are. Joel is going to keep working on this probably. We have somebody coming in June 14th and 15th. We have visiting IU for 2 days. We'll have Claire Bryant and Gary Ann, who are big experts in this kind of modeling. And so this work will be something that we can show them and discuss with them. And I think they'll be pretty impressed with how far you've gotten. There'll be always is more to do, but I think that this shows is really good. I mean, I'm a little sorry that we picked the Hoffman paper, because picking something simpler that would have been more replicable might have been better. And for maybe there aren't other necroptosis models because it's not such a commonly understood pathway. Certainly for apoptosis, there are published models that are simple. But I think this is fine.
Yeah. Yeah, I don't regret in any time getting his paper to reproduce. I think I learned a lot. We learned a lot with just trying to replicate something and seeing the challenges. We talked a lot about this, and you mentioning that paper that shows. It also opens up a conscious for all of us that are doing science to make something that as easy to reproduce as we can, since science is not done just by one, and we want to build on top of other people instead of like trying to do everything again, right? Starting from using the blocks that we have instead of making the block itself.
And I think your replication of Fiona's model, I mean, sort of ironic that would have been a perfect class project in the fall by itself. But that replication of Fiona's model is very nice. And By itself, that's already plenty. So.
Yeah, now I'm working on the integration with the CC3D because it's a little bit, I have to keep track of,
Right.
okay, what this means to the model in CC3D, right? And transforming everything now in spatial and saying, okay, how much this actually move or this gradient actually move in this environment is a different type of question. And I think we're probably going to dive in more into using abstract or AU, always forget. I always forget what the term means, but arbitrary units instead of real ones, because I think it would be very hard to find something in the literature that says, well, this molecule moves in the in this medium at this velocity.
But it's actually important to know that. So that's where, and actually this came up with Josh's presentation too. There was a sort of a quick comment early on where you said that you talked about patterning where you had long range inhibition and short range activation, Josh. But you said, but then the values of the parameters you gave were the opposite. You had short range inhibition and long range activation. And so the key concept always for diffusion is what's called the diffusion length, which is the square root of the diffusion constant divided by the decay constant. And that tells you essentially how far the signal propagates. Now, if it's time varying, there's also a temporal thing, which isn't included in diffusion length. But what you really care about is essentially what is the diffusion length compared to the cell diameter. If you measure diffusion lengths in units of cell diameters, diffusion length is one cell diameter, the signal propagates 1 or 2 cell distances. If the diffusion length is 10 cell diameters, it propagates 10 or 15 cell diameters. And so that's really the important thing, is that number, that ratio. And now to get diffusion lengths estimate, to get diffusion constant estimates requires a little bit. Sometimes you know, somebody notices that the signal gets 10 cell diameters away, or 1 millimeter away, or 10 microns away. Then you can invert, if you know the distance the signal travels, and you know what the diffusion length is, and then you can back everything out. If you don't know it, for molecules and even for viruses, there's a upper limit on the diffusion constant, which is actually what Einstein won the Nobel Prize for. He didn't win it for general relativity. He won it for solving the diffusion equation. It's called the Stokes-Einstein relation. And it's only approximate, but it says that if you have a molecule that is spherical in water, depending on how big it is, the diffusion constant is such and such. And that's an absolute upper physical bound on the speed of diffusion.
So it's related with the size of the molecule?
It depends on only two things: the size of the molecule and the viscosity of the fluid.
Of the fluid, okay.
And so the basic calculation isn't that bad. It's a little bit of algebra, but then it scales. If the molecule is twice as big, then you could change, then the diffusion constant goes down by a factor of the radius to a power. If the viscosity is 10 times bigger than water, then the diffusion constant is 1/10. It's a linear scaling. And so you only have to calculate that number once, and then you say, gee, my fluid is 10 times as viscous as water, so the diffusion constant is 10 times lower. My particle is 10 times bigger, therefore the diffusion constant is 100 times smaller. And so they're actually, if you look up diffusion constant, diffusion length calculator, or Stokes-Einstein calculator, you'll find online things where you type in the size of the speed, the size, and it'll give it to you. You can even type in the molecular weight and it'll assume the protein is roughly spherical, and it'll give it to you, because chemists do this all the time, really all the time. Now, it's not exact because molecules aren't perfect spheres. They're not even perfectly compact. Fluids aren't perfectly Newtonian, and so on. But it's the fastest that you can possibly have diffusion by itself. Now, in the lungs, for example, there's also flow, and so then things move faster than that. But Stokes-Einstein gives you at least a sanity check. Is the influence 10 microns, 1 cell diameter, 100 microns, 10 cell diameters, a millimeter, 10 millimeters? And that order of magnitude helps you a lot, because it tells you, is the signal propagating over the whole tissue? Is the infection in one cell going to affect the whole lung, or just the local alveolus, or just something else? And so those numbers are put. The diffusion constants are actually pretty high, which is a pain for CompuCell, because it means that you have to run a lot of diffusion steps, so your simulations are slow. So when we simulated viral replication, our viral diffusion constants and our interferon and interleukin diffusion constants were too slow. And so those are, but you can get estimates of those. In fact, in Josh's paper, he did do those estimates. And so you can get an idea. And there's this, there's a BioNumbers database from Harvard. And there's another one from Phillips at Caltech, which have lots of really useful reference numbers that you can look at. Again, this is a little bit more biophysicsy than we do in this class so often. But you can at least know. For example, a virus typically has a diameter. A bacterium has typically a diameter of a micron. A virus typically has a diameter of a tenth, less than a tenth of that, a 20th of that. And that means that its diffusivity is going to be at least 1,000 times bigger. But a small molecule like interferon or interleukin, it will have a diffusion constant 10 times or 100 times that of the virus. And so then you can get at least the ratios approximately.
Yeah, because one of the ideas that I have for something to do on NanoHub was actually play with that, but play with how much the diffusion of one in comparison with the diffusion of other, and which results we could get. But that you just said, that information give us a bound, like not a lower bound, but a top bound, because we know that it cannot be bigger, it cannot be faster than that calculation, right? We can always go slower than that, but not faster. So it's a good tip.
But it also depends. I mean, the classically you do the calculation in water, but the viscosity of almost any biological fluid is much higher than that of water. And so if you look, for example, at the mucus in the lungs, the viscosity is about 1,000 times that of water. So the diffusion constants are about 1,000 times smaller. And that saves you, because if you actually had to do all these calculations in water, the time step you'd have to use in CompuCell would be so short. It would be like poor Connor and his it'll take you 10 years to run the simulation. The reason there's the steady state diffusion solver in CompuCell, which we don't use very much, is for dealing with these situations where the diffusion rate is so fast compared to everything else that you might as well assume it's equilibrated. It's still slow, but it buys you some, it saves you some time. It's also a good argument for putting all the diffusion solvers on CUDA, so that again, for the kinds of things we typically do in class, we use relatively low diffusion constants, and so things run in a reasonable time. But oxygen, in particular, oxygen, glucose, ATP diffuse really fast, and so to actually simulate the diffusion of those in a time-varying way realistically is very computationally expensive. If your classic Monte Carlo step is 10 seconds, it's okay. But if you're simulating a year's worth of a person's life and use a Monte Carlo step of a minute or 10 minutes or 15 minutes, then the diffusion length over the diffusion distance over those periods is large. And you have to, it winds up, it's expensive to do the time dependent calculation. But again, those are fascinating things. If there were more people interested in this, I'd give a second course on the topic. But it's there are a whole series of really in each one of the projects has brought up interesting questions, whether it's theory of evolution and how do you do things, how do you do these complex adhesion, how do you optimize code. Each project has explored interesting new things. I appreciate all of them. So again, I don't think, next week, I would like people, if everything is the same, I suppose you don't have to present the same things exactly the same. If your presentation be literally identical, I guess you can say that. But maybe there are things that you could add for people today, I made some notes about maybe you could give a little more biological background or explain this or that. So I'll send those around. I do ask you each to send out your comments to each other, because I think it's important to have that opportunity. And I'm happy to work with people if there are code issues or otherwise to finalize projects. That would be fine. But I think everybody has, even if things aren't perfectly finished, I've had semesters where at the end of the semester somebody said nothing worked, and that's a tough one. Because while I appreciate effort, ultimately there has to be something to show for the effort. And so I think everybody, even if nobody has exactly what they hope to get done, everyone has achieved something. And I hope people have learned something from that too. I was hoping to do that delta notch, but I think people probably don't feel like doing a delta notch exercise at this point. No. I'll post the slides, although they're not very different. Maybe I'll show you just one slide. The key thing, and I showed it last week, but we didn't really go over it. The key thing with delta notch, and the reason you ran into this trouble, and I realized it was in the slides, is there's this very simple model for activated delta and activated notch. And what I asked people to do was, I give you the algorithm here, so I give you pseudocode for it. And I'm sorry, I don't know why the formatting is messed up. It was okay when I showed it a few minutes ago. What I had, and this simulation is available. The simulation code should be available as an example. Actually, I think it's a CompuCell demo, as a matter of fact. It's a delta notch demo. But the only thing that the place where you got stuck was that this very simple model here exists, and this is a change in the order in which we taught things. In the old days, sometimes history is a burden. So initially, CompuCell could load SBML models, but we didn't support the antimony models. And the problem with SBML is that you can't edit, it's not human readable easily. And so you had to edit it in another editor and then read it in as a file. And so then when we created the antimony support, we did SBML first and then antimony because we had SBML before. But in fact, there's really no reason to teach SBML model import, although I have it in the slides, because the antimony is so convenient. However, in those days, since you couldn't write these things in antimony, what we had was we had people read in an SBML version of this model, this very simple model, because it's published. And then that request that was lurking in the homework, it's an old homework problem, was you read it in from this online repository, you converted it into antimony so you can see what it looked like. But in fact, it's exactly 2 lines. So writing it by hand takes all of 1 minute. And so I really do apologize, Alex, for putting a mine under your feet. But in the slides, and the slides are there from last week, the code you need is here. And so the only purpose of that was to take, if we jump, and actually the full code that you need for the whole thing is here in the slide deck. But in the example that I had here at the end, which is the cyclin example, you start with this Tyson and Novak model, and then this is showing you how to download it. So there's this nice library called Biomodels that has all these models in it. The Tyson model, cell cycle regulation, exists there. You can download it and put it into a cell, and you can actually, CompuCell will actually allow you to pull it off of the web if you want to. And I think, I'm trying to see if I have, no, I don't. I took out the slide. You can see the XML expansion. It looks like MuParser, or it takes, I mean, not the MuParser, but the output of MuParser. So you'll see each operation in that code will generate, there'll be a species definition that there'll be each operation, that code will generate a stack of XML tags. And you can walk through it. It's not the end of the world, but it's not much fun to read or write. And so that's why the antimony is more convenient. So I do apologize. But if you want to implement this, if you want to implement the that delta notch, it's all of the code you need is in the slides from last week. And I do apologize again for having left in that you should turn the SBML back to get this code, because it's really not necessary. So, okay, well, thank you, everybody. Please get me your comments. Please meet with each other and do more your little intensive review and get those to me so they can be circulated. And I will meet with you again in a week to see how things are finalized. And again, if in a week you come back and say it's the same, that's okay. You certainly don't have to repeat everything you said this week. On the other hand, if you want to give the whole talk again, you're welcome. Have it online. And I appreciate everybody's work during the semester. And I do think it was very creative. Everyone was creative, came up with neat ideas. Nothing ever works exactly the way you expect, but I hope it was a good learning experience. So thank you again for sharing today, and I will see you in a week. And if people want to talk about things between now and then, let me know, and I'll make myself available. Okay. Good night, everyone.